Welcome to Module CCS 16 on the fundamentals of cleaning. This session will discuss the fundamentals of cleaning in the pharmaceutical and biopharmaceutical industries. In today's session, we will cover the importance of cleaning in preventing and eradicating microbial contamination in pharmaceutical and biopharmaceutical environments. More specifically, we will look at the foundations of clean in place, clean out of place, and parts washer systems, the differences between manual, semi-automated, and automated processes, the selection of cleaning agents, and equipment, operational, material, and product risk considerations. A healthy human eye can identify particles larger than 40 microns, about half the thickness of a hair. Most harmful particles are invisible to the naked eye. It is impossible to determine if a surface is clean just by looking at it, making it challenging to clean an apparently clean surface. This is akin to painting with invisible ink. Particles smaller than what the human eye can see remain undetected in the surrounding air, making it impossible to visually identify airflow patterns inside a cleanroom. Cleaning is one of the most contaminating activities in a cleanroom. Cleaning a cleanroom is vastly different from domestic cleaning. The ISO 14644 standard provides essential guidelines that highlights the difference. First, it classifies surfaces based on their criticality. These can be critical surfaces, general cleanroom surfaces, other surfaces. So this shows that not all surfaces are equally critical. Secondly, the standard defines 3 levels of surface cleanliness: gross cleaning, intermediate cleaning, and precision cleaning such as disinfection. The operational parameters for the cleaning process include cleaning agent, concentration, contact time, and temperature. Factors affecting the cleaning process are the product characteristics and the product conditions. Specifics about the cleaning equipment include flow rate during each step and the sequence and pathways for manual or automated cleaning steps. Establishing effective cleaning processes in pharmaceuticals involves 1. Identifying control variables, cleaning measurements, and performance criteria. 2. Conducting lab experiments to establish control ranges for cleaning parameters. 3. Considering critical process parameters and critical quality attributes of the cleaning system during design. Cleaning processes are composed of multiple steps. Each step has a specific function with set parameters to ensure effective soil removal. Exact details vary between sites and type of equipment, as each has its own unique cleaning process tailored to suit its function. The TACT framework defines effective cleaning in pharmaceuticals. The first T stands for time, which is duration of each cleaning step, measured directly using cycle step timers. The A stands for action, which refers to the mechanism used to apply the cleaning agent, such as soaking, scrubbing, impingement, or turbulent flow influencing how well contaminants are removed. The C stands for concentration, which refers to the strength of the cleaning agent, which in turn affects the cleaning performance of the agent. The second T stands for temperature, which can enhance the effectiveness of cleaning by increasing residue solubility and reaction rates. These parameters should be carefully defined with established acceptable ranges during cleaning process development. Equipment may be cleaned at its installed location, or it may be disassembled and moved to a central location for cleaning. In-place cleaning takes place in the equipment's permanent location. This can be automated using built-in systems, often referred to as clean in place or CIP, or it can be manual where personnel use brushes and hoses. Solvent reflux cleaning involves boiling a volatile solvent in a reactor vessel. These vapors from the solvent rise and condense on cooler surfaces within the reactor, which effectively dissolves residues. The residues are then soluble within the solvent itself. This is typically used for small molecule active pharmaceutical ingredients, or API, manufacturing through a process known as organic synthesis. Placebo cleaning involves running a placebo batch through the equipment to clean it of drug residues or process residues from the previous batch. This form of cleaning is typically used for highly viscous ointments and products which can be difficult to fully remove. Thank you for watching this introduction to this module. If you'd like to watch the remainder and get 25% off our Contamination Control Fundamental program, head over to our website at pharmalliance.ie/training-programs and use code YT25OFF. Please do it now as spaces are limited and it's expiring soon. Your time, and see you on the next module.